Good afternoon, ladies and gentlemen. I'm Victoria Harden. I'm the NIH historian and the director of the Stetten Museum, and I want to welcome you to our annual symposium presented by the DeWitt Stetten Jr. Memorial Fellow in the History of Biomedical Sciences and Technology in conjunction with NIH scientists with whom he or she has been working during the year. I wanted to give you just a brief background for those of you who may not know how this program got started. The Stetten Fellowship was created as a living memorial to Hans Stetten, Dr. DeWitt Stetten Jr., who founded this museum at NIH. And it has been an extremely successful program, in my opinion, in bringing together historians at the beginning of their career and working intramural scientists. One of the things that concerned me as we set this up was how little well known the intramural scientific program at NIH was, and one of the goals of the program was to bring to the attention of historians of science the work that was being done here in Bethesda, and then in the other direction to have historians help to explain to working scientists why it was so important to document their work beyond the published record. And I think both of these goals have been advanced in the years of the fellowship. This year, our fellow has been working in the National Institute of Diabetes and Digestive and Kidney Diseases, and I'm extremely pleased that NIDDK sponsored the Stetten Fellow this year, and especially to our 11, who has been the scientific director in charge of the fellow and who is my co-host for this event today. I also want to extend special thanks to the other two speakers, Drs. Gary Felsenfeld and David Davies, who have been the laboratory hosts for our fellow today. I also want to say that we decided we'd take some of the pictures in advance. That's why we were doing this, so that when we are finished, we'll all be able to adjourn rapidly to the reception, and you all will have a chance to talk further with the speakers. This brings me then to the pleasure it gives me to introduce the Stetten Memorial Fellow for this year, Dr. Boom Soon Park. Dr. Park was trained as a chemist and a historian at Seoul National University in Korea before he received a doctoral degree in the history of science at Johns Hopkins University in 1999. And we're also pleased that his mentor, Dr. Owen Hanaway, down here from Johns Hopkins, is here to hear him speak today. Dr. Park wrote his dissertation on the history of quantum chemistry, examining the disciplinary interplay between physics and chemistry in the 20th century. He has published several papers on this subject. He has been the recipient of fellowships from the International Rotary Foundation and the American Philosophical Society, and he has received research grants from the American Institute of Physics and from Oregon State University. Dr. Park joined the NIH this past year as the Stetten Memorial Fellow, and since then he has been investigating the characteristics of NIH intramural laboratories in general, and the Laboratory of Molecular Biology at NIDDK in particular. He has been awarded a second year for this program by NIDDK, and during the second year, Dr. Park plans to work on the discovery of the triple helix structure of polynucleotides in the late 1950s by Alexander Rich, Gary Felsenfeld, and David Davies. Dr. Park is also interested in investigating the role of James Shannon and NIH in nurturing biomedical research manpower in the United States in the 50s and 60s. Today, he will speak to you on the most intriguing title, More Academic Than a University, Three Freedoms, and the Laboratory of Molecular Biology in NIDDK. Dr. Park.
I'd like to start my talk by giving you an explanation of the symposium title. Since I came to Bethesda last year, I have been interested in the characteristics of NIH as a place for research. I looked for sources describing scientists and laboratories here, and I came across some comments made in outside reports. One was the report to President Lyndon Johnson in 1965, submitted by the Special Committee for Investigating the Operation of NIH. The committee had numerous on-site visits before giving an intriguing observation on the intramural program. The NIH scientist has at least as much, and probably more, academic freedom than his university counterpart. The report went on to describe the NIH scientist chooses his own research project and determines his own direction of approach. He finds it relatively easy to secure modern equipment. He has fewer distractions to keep him away from his laboratory, faculty meetings, committee activity, and the like. Not being in an educational institution, he need not teach. He can devote all his time to research. This image of NIH scientist enjoying academic freedom persisted. In 1988, the Institute of Medicine examined the NIH intramural program, describing its characteristics with the phrase, 3 freedoms. Freedom to choose research topics, freedom to devote all working hours to research, and freedom from the need to develop grantsmanship skills. Of course, the NIH is not a perfect place. I could also find intramural scientists complaining about government regulations, limitations on traveling, slow procurement, and less competitive salaries, especially for medical doctors, are among the sources of complaints. But there is no doubt that a number of scientists came to and stayed at NIH because they liked the research environment here. When did NIH become an attractive place for research, and how? Today, I'm going to examine the origins of the unique research environment at NIH in the broad post-war context, taking a close look at the establishment of the Laboratory of Molecular Biology at NIDDK. What I want to stress is the role of intramural people, that is, directors, scientific directors, and lab scientists, in securing research freedom and creating an academic atmosphere. Let me clarify my point with this diagram. The NIH is, as you know, an invention by public health reformers, politicians, bureaucrats, and scientists. For example, the fact that the names of institutes have changed so often is a reflection of social and political pressures. But these pressures are much diminishing as they pass within NIH. If I use a biological metaphor, directors form a semi-permeable membrane, which selectively allows the passage of outside demands. The scientific director of each institute forms another semi-permeable membrane. Perhaps the lab chief makes a third membrane. In this way, bench scientists may feel only an intellectual pressure that they have to show that they are doing excellent and productive research. I would argue, therefore, that the unique research environment at NIH is an invention by these intramural people. Let me start with the broad context and directors. It is now well known that the NIH emerged as a major patron of biomedical research in the years following World War II. Historical circumstances for the post-war transformation of NIH were complex, but the most notable thing was that a social consensus was made to support research in chronic diseases. The NIH became plural with the creation of new institutes bearing names of disease, and changed the focus of research from infectious diseases to chronic diseases. I'm not going to examine this development in detail, but I want to say one thing, that the disease category system never discouraged basic research. The relevance of research to disease was broadly interpreted, and NIH directors sought to avoid the pressure for applied research at the sacrifice of basic research. For example, James Shannon, who directed NIH in the years between 1955 and 1968, pointed out, "A primary aim of the NIH is to administer the program in a way that prevents the potential hazards of the disease category system from becoming real." With a practical and efficient application of the disease category system and with strong support from Congress, Shannon led what we now remember as golden years of NIH. In the 1950s, the morale of NIH scientists was high. According to a survey, they considered their place better than other government research institutes and just as good as the best universities, hospitals, and industrial laboratories. In general, they found NIH free from the restrictions one might have expected in federal laboratories. In fact, the initiative to create an academic atmosphere at NIH did not always come from above. Scientists found their ways to exchange ideas, information, and materials. For example, Arthur Kornberg, Leon Happel, Bernard Horecker, and Herbert Tabor formed a small luncheon seminar group in 1946. They met every day, including most holidays, and discussed a wide range of subjects in biochemistry and enzymology. More and more people joined this luncheon seminar, and it also served as a place for informal presentations by outside scientists. Similar seminars or journal clubs started spontaneously in other disciplines too. In addition, the early 1950s saw the beginnings of evening classes offered by NIH scientists for themselves. Scientific directors in charge of the intramural program added more fuels to this self-teaching effort. In 1959, together with some scientists, they established a non-profit, non-governmental educational organization, the Foundation for Advanced Education in the Sciences. The FAES took up the administration of evening courses and also started a PhD program in conjunction with the Johns Hopkins University. Further academic features, such as the bookstore, the concert series, and lectures in non-scientific fields became available. Scientific directors were also concerned about improving the social status of the scientists at NIH. They imported principles of tenure and sabbatical leave and established the faculty center. They even organized the Assembly of Scientists to facilitate the communication among scientists and take part in the decision-making process. Its impact was not as great as they expected, but this kind of faculty organization was unprecedented within the federal government. With all these innovations, the Bethesda campus looked more like an academic campus. Dewey Stetten Jr. was among those who sought to improve the quality of life at NIH. He came in 1954 as scientific director of the National Institute of Arthritis and Metabolic Disease, or NIAMD, a former name of NIDDK. By that time, Stetten had become a well-known biochemist, especially for his gout research. Taking an administrative position, he also proved himself a man of vision and energy. Stetten clearly saw that the key to the success of NIH intramural program was to recruit competent investigators and let them work with as much freedom as possible. In 1956, he said, "The greatest return will be secured if the mature scientist is allowed and encouraged to select the problems on which he will work. It is our belief that the meritorious and experienced investigator will in general be the wisest judge of his field of endeavor. The most important function of the scientific directors, therefore, is in the selection of senior scientists, in their encouragement, and in the attempt to procure for them facilities." Stetten, being in a position of scientific director, knew where promising talents were. Furthermore, as a leading biochemist, he could point where the research front was. At the time, molecular revolution in biology was well underway. The DNA structure was discovered in 1953, and the so-called central dogma regarding the flow of genetic information from DNA to RNA to protein was conceived. A number of scientists in the world were jumping into the study of polynucleotides and proteins. Stetten felt the need for encouraging and supporting more research in this field. But the problem was that laboratory space for NIAMD was already approaching saturation. Stetten found a golden opportunity in 1960 when the National Institute of Dental Research decided to move from Building 2 to Building 29. He wasted no time claiming the available space in Building 2 and drawing a plan for a new laboratory. Stetten sent a proposal to the director of NIAMD. The exploration of biological events at a molecular level has become an increasingly important area of biomedical sciences over the past decade. This is reflected in the establishment of journals and of a new society dedicated to the field of molecular biology. In order that NIAMD may make its proper contribution to this burgeoning area of research, it is proposed to assemble talent from within and from outside our institute into a new laboratory, tentatively to be named Laboratory of Molecular Biology. Stetten appointed himself acting chief of the lab, an unusual move for a scientific director. He then put together a group of competent scientists. Within NIAMD, Gordon Tompkins and his section on metabolic enzymes in Leon Hepper's Laboratory of Biochemistry and Metabolism moved into a new space. Along with Tompkins came Todd Miles and some others. Bruce Ames and Robert Martin also moved from the same lab to work in the section on microbiogenetics. The section on chemical genetics was built around Harvey Itano from the Laboratory of Pathology and Histochemistry. David Davis and Martin Gallot from the National Institute of Mental Health were assigned to the section on molecular structure. Finally, Stetten recruited Gary Felsenfeld from the University of Pittsburgh, who would lead the section on physical chemistry. In 1960, molecular biology was a new science. It was a new way of examining information flow in a cell. It could be seen as a genetics using biochemical techniques or a biochemistry studying genetic mechanisms. In short, molecular biology was not so much a discipline as a multidisciplinary approach. The newly established Laboratory of Molecular Biology demonstrated this point very well. Scientists there had diverse educational backgrounds, ranging from enzymology to quantum chemistry. Some had received no formal training in biology, and others had never touched an X-ray machine before. Despite this diversity, however, scientists in the lab could maintain a high level of mutual inputs. How was it possible? Without a doubt, self-teaching devices at NIH, such as luncheon seminars and evening classes, had given opportunities to learn basic and advanced knowledge in various disciplines. In the Laboratory of Molecular Biology, scientists also created their own weekly seminar. Each week, scientists crammed themselves into the sloped roof attic meeting room where they would present their papers. Brad Thompson, who joined the lab as a clinical associate in 1964, recalled that this weekly seminar was clearly a challenge because of the scientific rigor with which the data were evaluated by the group. In the Laboratory of Molecular Biology, reading an article was considered as important as pipetting a liquid. Creative thinking and diligent work went hand in hand. This may sound too obvious today, but it was not necessarily the style of other labs in the 1960s. Martin Gallot has described his experience at Stanford. He was on sabbatical leave And since he was trying to generate ideas, he would spend a fair amount of time during the day reading journals. And a couple of times, he had the experience of somebody coming in and looking at him reading. They were puzzled and then said, "Oh, that's right, you are on sabbatical." Gellert and many others credited Gordon Tompkins for establishing the style of the lab. Tompkins became the lab chief in 1962, but his influence was not through this position. Tompkins was indeed an extraordinary person. He was an MD, a PhD, an expert in hormone studies, and a jazz musician of professional quality. His knowledge was astonishingly diverse, and his memory was simply legendary. But his great talent was in the realm of communication with his fellow scientists. Henrik Eisenberg, a visiting scientist in 1965, has described, "I found the LMB and Building 2 to be a most unique place of research and human interactions. Much of it I ascribe to personality of Godi, whom one could not pass in the corridor or near the coffee pot without a fruitful scientific exchange or a humorous but profound comment on political, social, or cultural events." Stetten also said, "Never have I known another who could exceed him in his ability to stimulate the interest of colleagues in scientific problems. All of those who had much contact with him were in his intellectual debt." It might be difficult to copy Gordon Tompkins's wit and charm, but his enthusiasm for new ideas was certainly contagious. At the time, as Martin Gellert has said, the intellectual life of the laboratory was in the corridor. As you can see, the corridor was short and narrow. Anytime you went out of a room, you could start up a conversation with somebody. The laboratory was new, people's ultimate directions were not firmly fixed, and so there were many things that could happen with just informal conversation. Informal conversation often led to scientific collaboration across sections, and if necessary, the number of collaborators could grow. People were willing to lend their hands and share their ideas. They were strongly independent-minded, but they were equally open-minded. Their intellectual openness also attracted a number of collaborators from other labs. Like the collaboration inside, the collaboration with outside scientists initiated informally during a phone call, at a luncheon seminar, or during a game of tennis. Intellectual freedom and organizational flexibility have been virtues at NIH. David Davis has said, "One of the things that really attracted me to this place was that there was no attempt to coerce anyone into working in any particular area, but the scientists decide themselves what they wanted to work on and what was interesting." Martin Gellert has also made a perceptive observation. "My feeling is that it is probably easier to get help and collaboration here at NIH than it is in most universities, partly because professors in universities have large research groups, and partly because their fundraising capability depends on their reputations. University professors tend to be a little more separated and to maintain the integrity of their own research group." Ed Roll, who spent many years as scientific director of NIAMD and deputy director for science at NIH, has also pointed out, "At the NIH, getting support was not a zero-sum game. If you get something, it does not mean that the guy next to you won't get it. If you get promoted, it is unrelated to whether the guy next door gets promoted. This cuts down on rivalries and jealousies a little bit." To be sure, the funding structure for the intramural program contributed to making a collegial atmosphere in Bethesda. But scientists in the Laboratory of Molecular Biology were more than colleagues. They were friends. They saw each other socially and knew each other very well. For some, friendship had begun even before coming to NIH because of their common training at Caltech as either graduate students or postdoctoral fellows. For others, mutual respect and trust developed during daily contacts. The major change of the lab took place in the late 1960s when Gordon Tompkins, Bruce Ames, and Harvey Eternal left for the university positions. Around that time, Todd Miles, Martin Gallot, and Robert Martin became new section chiefs, and all section chiefs agreed to invent a novel system of a rotating lab chief. Each section chief took a turn as lab chief for a year at a time. Gary Felsenfeld has said, "We governed the laboratory. We still do as a group. We always managed everything very well. We never asked for anything foolish, so people trusted us. And over the years, we have had an idyllic existence in terms of being able to focus on science, being trusted to choose whatever was the best subject, and being supported." They may have enjoyed a sort of idyllic life in the federal government, but no one has questioned the productivity and quality of research out of this lab. One independent measure of success can be membership in the National Academy of Sciences. There are 15 members among people who have stayed in or passed through the lab. Furthermore, their papers have repeatedly demonstrated that basic research would finally pay off. Let me give you a few examples. Here is Dr. Davis. David Davis's study of protease in the 1970s served as a paradigm in the following decades for understanding HIV protease and developing its inhibitors. In fact, it was Davis who convinced the NIH administrators in 1986 to use Director's Discretionary Fund for launching a program in structural biology. None of the virologists at the time were thinking about the virus at atomic resolution. But Davis recognized that it was important to start immediately to try to solve the structure of HIV proteins as targets for drug development. We can find another example in Martin Gallot's discovery of DNA gyrase, an enzyme that changes the orientation of spore coiling of DNA. He discovered it in the 1970s, and it turned out that DNA gyrase could be the target of several antibiotics. For instance, gyrase inhibitors like ciprofloxacin are now widely used to treat urinary tract and other infections. Of course, Gallot did not become a millionaire out of his research. Gary Felsenfeld's study of chromatin and the organization of DNA in the nucleus gives a third example. He has been working on this subject for many years, and now chromatin turns out to be the basis for gene expression in higher organisms. We cannot emphasize too much the significance of this knowledge in biotechnology. All of these examples strongly suggest that the long-term investment and the trust in mature scientists have been well justified. Writing his autobiography in 1983, Stetten was proud of the lab. Stetten said, "This cluster became one of the hot spots of research on the NIH campus, and I have always regarded its establishment as one of the more positive contributions that I made to NIAMD and to NIH." Stetten created the lab, and the lab scientists invented their own research environment, tradition, and reputation. They have used wisely and responsibly 3 freedoms available at NIH. In conclusion, I'd like to say something about the picture in the symposium poster. This is a 16th century painting by Peter Bruegel. Here, a whole town is given over to the children as a playground. They are playing many kinds of games. See, rolling hoops, riding hobby horses, tug of war, playing with tops at the bottom, and the like. They are playing alone, in a small group, or in a big group. No one directs them to play this or that game. They are free, self-directed, grouping and regrouping. While playing together, some of them become friends. This painting is my visualization of the NIH campus, and especially scientists in the Laboratory of Molecular Biology. Thank you.
Before you take off that picture, Dr. Felsenfeld pointed out that that's a pretty reasonable rendition of Building 2 before we move to a little more spacious quarters, Building 5. I'd like to now, it's a real pleasure to now introduce Dr. David Davies, one of the world's foremost crystallographers, whose many postdocs passing through his lab have achieved prominence and are established in a vast number of laboratories around the world. And it's good to point out that David is not only is an expert and shows his devotion to crystallography, structural biologist, but he has devotion to tennis. We saw that Prince racket that he was holding there, sailing, and azaleas. And I'm not too sure which order these are. I must share one comment, if David will allow me. David received his PhD in physics from the Magdalen College at Oxford. Well, there's my imperfect accent. In early 50s, 1952 or so, and PhD in physics. And he shared with me once that his mother really wanted an MD in the family. And, you know, the biblical injunction is do not go against the wishes of particularly your mother. And I think for the good of the science, David showed his familial independence, and I think we're better off for all of that. So, David, please.
Thank you very much. I really enjoyed hearing about the early days from a different point of view. And Boomsan has asked me to say a few words about the development of structural biology in the laboratory of molecular biology. But first, I'd like to say that one of the very good reasons for the success of the lab was that we really had fantastic scientific directors who supported us, who stepped in at moments of crisis, and who really gave us a lot of support. And I was very happy to see Ed Rall here right now. We had Hans Stetten, Ed Rall, we had Jesse Roth, and up until very recently, Alan Spiegel. And I think we were very fortunate in that respect. Let's see. Do I control this? Stray, focus, stand by. Could I have the first slide, please? There's the... Now, one of the things that Boomsan pointed out was that the lab had a very strong Caltech background. 4 of the 5 initial section chiefs were all together at Caltech at the same time. And moreover, 3 of them, Harvey Atano, Gary Felsenfeld, and myself, worked directly with Pauling. Gary was Pauling's graduate student and wrote a thesis on a theory of ferromagnetism believe it or not. And Harvey was the person who, with John Singer, showed that sickle cell hemoglobin was indeed different from normal adult hemoglobin, and thus establishing it as a molecular disease. And I worked on the precise structure of small molecules related to peptides and 2 amino acids, which were used by Pauling for things like the alpha helix and the beta sheet. So the early people in the lab had a very strong molecular background coming from Caltech, and in particular coming from Linus Pauling. Now, to put some of the structural biology in context, how do I set the, this must be it, yeah, way forward. This is a slide of the structure that I worked on as a graduate student at Oxford. It's basically a 12-atom small molecule, and this was in the days before computers, and it took well over a year to determine this structure. We had to use something called Beavers and Lipson strips, and you had to use adding machines, and it just was a tremendously complicated and tedious job. There were no data collection equipment. You had to measure all the intensities visually, and it seemed to us at that time that the probability of being able to solve a protein structure with many hundreds, if not thousands of atoms in the structure was out of the question. But during the 1960s, computers developed. During the 1950s, computers developed, and to the extent where there were 1 or 2 really significant computers around. One of these was in Cambridge, EDSAC-2, and John Kendrew was able to use these with his colleagues to determine the structure of a small protein. This happened about 1960 or so, and this slide shows how primitive things were. I mean, we constructed this forest of rods, and John is actually, and we put in colored clips to represent the electron density, and John is actually building in, as you can see, you could directly visualize an alpha helix, and John is building in the alpha helix into this forest of rods. A very, very slow and tedious job, a job that took up, just like the computers in those days, a tremendous amount of space. And so that if you were to build the structure of a ribosome or something like this on this scale, you would require the whole of this room in order to be able to do it. So what Kendrew showed was that protein structures were, in fact, doable. And he led the way for the rest of us. Now, what's happened since then, could I have the next slide, please, is that there has been a tremendous growth in the determination of protein structures. And this slide illustrates the deposition of protein and nucleic acid structure coordinates in the protein data bank starting in 1972. And as you can see, for a long time, there were virtually no new structures. Maybe about 10 or so new structures by about 1972. And gradually, they increased, certainly by about the mid-80s, they began to take off, and the red shows the new structures each year. They're now up to about 3,500 new coordinates each year, and the green shows the cumulative total. And so a lot of this is due to the expansion of the field. A lot of it is due to the development of computers, so that it's now possible to do things that you couldn't even dream of doing 20 or 30 years ago. And a lot of it, next slide please, is due to the development of new X-ray detection devices and also new sources of radiation. For instance, there are now synchrotron sources of radiation. Here's a synchrotron ring, usually in government labs. This one's at Grenoble, And in a very nice situation, I'm told that there's a tremendous queue to get there when during the winter months when skiing is very popular. And you see the size of the ring, and these machines now are capable of producing X-rays that are hundreds or thousands of times greater than you can get from your laboratory machine, and also are tunable so that you can select whatever wavelength you want in order to be able to determine the structure. The result is that now there are something like more than 50 new structures being reported every week. And so much so that it's impossible for the individual to keep up with this anymore. And it's going to lead undoubtedly to the development of biocomputing in order to be able to process and analyze and keep up with all these data. Okay. Now, I've been asked to talk about the present state of structural biology in LMB, and the next slide shows the major people involved. We have 4 separate research groups. There's Wei Yang, who's interested in DNA repair and recombination, Fred Dider, excuse the typo here, melatonin synthesis and recombination, Jim Hurley, signal transduction and genomics, and myself. I'm interested in a variety of different things. Next slide, please. I should say, could you go back one? I should say also that we have Eduardo Padlan, and Eduardo's interested in therapeutic antibodies. He's a distinguished crystallographer, and but he also likes to maintain a very low profile and didn't want his name put on this. And we're also in the process of hiring a membrane protein crystallographer to fill what we perceive as a gap in our present arrangement. So here's Wei, here's Jim Hurley, here's Fred Dider, and myself. So what I'm going to do now is just show you 8 slides, 2 each from each of these people, to give you an idea of what they're doing. First, Wei Yang. Wei is working, her work principally over the last 4 years or so has been in DNA mismatch repair. The mismatch occurs, it's recognized by, there are 3 proteins involved, mut-S, mut-H, and mut-L. It's recognized by mut-S, but of course mut-S doesn't know on which strand the mismatch occurred, so the parent strand is recognized by methylation opposite a particular sequence, and is recognized by mut-H, and mut-H is an endonuclease, and when mut-S recognizes the mismatch, then mut-L gets activated, and in turn activates mut-H, and mut-H then cleaves, strips off the daughter DNA up to the point of the mismatch, and then that's repaired by various ligases and things to give you, hopefully, the mismatched DNA. Wei has worked on the structures of all of these. She's worked on mut-L, she's worked on mut-H, and she's also worked on a complex of mut-S with a mismatched DNA, and that's on the next slide. And to show you the complexity of the structure, here's the structure, it's a dimer. There is a tunnel through which the DNA goes, and it's possible from this to see how the DNA recognizes the mismatch, and it's possible to see how it activates the ATPase activity, which the ATPase site being located up in this part of the molecule. Next slide, please. Fred Dider is working on serotonin transacetylase. This is a slide of the structure. This is an enzyme that creates a precursor to melatonin, and here he's got CoA complexed to a serotonin-like molecule, and it's the first structure of this kind of enzyme, which belongs to the general superfamily of the histone transacetylases, and it's the first structure of these in which you can see the binding site of the donor as well as of the acceptor of the acetyl group. The next slide shows that he is also interested in recombination, and this is the structure of, let me get all this straight. This is the structure of HP1 integrase. It's a site-specific integrase, and it's quite different from the, say, the retroviral integrases. It's an integrase that uses a tyrosine residue to form a covalent intermediate with the DNA. And what Fred's structure showed was that this tyrosine is in fact cis to the molecule containing the active site. It also shows the role of the C-terminal helix swapping in the oligomerization of the enzyme. He's also determined the structure for a TN7 transposase. Next slide, please. Jim Hurley has had a number of successes. He's worked on the structure of the adenylate cyclase. It's remarkable that this structure took so long to be determined. I had assumed that it had been determined years before. This is the structure of the homodimer of the C2 domain of the adenylate cyclase. The actual active enzyme contains a C1 domain and a C2 domain, but the C1 domain is very similar to the C2 domain, and for reasons of being able to crystallize it and so on, he worked on the structure of the C2 homodimer, and it shows the role of forskolin, which is an activator of this enzyme, in joining the two subunits together, the two domains together. The next slide shows his other major interest, which is in the phosphoinositol sort of second messenger system, and phosphatidylinositol second messenger system. And he's worked on a number of these structures, phospholipase C, PIP kinase, and then various domains, the C1 domain and C2 domain of phospholipase C, and of protein kinase C, the 5 domain, and then these others were worked on by other groups. That's his major interest, and he's still going strong on all of this. My own interests are shown in the next slide. One of them has been a long-standing interest. It's a collaboration with Edith Miles, also in NIDDK, on multi-enzyme complexes, and in particular on the structure of tryptophan synthase, shown here. This is an alpha enzyme and a beta enzyme joined together, and the alpha enzyme makes indole, and the beta enzyme takes indole and serine and makes tryptophan. And it's the last two steps in tryptophan synthesis in bacteria. And one of the interesting results that came out of this was that very early on, in the days of Charlie Janowski, he showed that there was channeling of the indole, that the indole never really appeared as an intermediate in the reaction. And what we discovered was that, and this was the work of Craig Hyde originally, that there was a tunnel that runs from the alpha-active site to the beta-active site. And this tunnel provides a means for free diffusion of indole from the first active site to the second. And the reaction at the second active site is so rapid that as soon as the indole is made and everything is all set up, and you have an available group at the second active site, then the indole is used up immediately. There are all sorts of flaps that open and close, and there are flexible domains that open and close, and there are allosteric interactions between this site and this site, that ensure that you get efficient catalysis and that the indole is not lost out into solution once it's been created. And finally, we have another collaboration, the last slide please, with Bob Craigie in LMB, and we've been working with Bob for about 10 years now on the structure of the HIV integrase. And we eventually focused on the catalytic core domain, and the object has been to try and understand the mechanism of action of the enzyme, and also to try and find inhibitors that would bind in the active site that would provide could act as lead compounds for further drug design. HIV integrase is the third enzyme of HIV. There are inhibitors for the protease and for the reverse transcriptase, and it seems like a very attractive target for further drug design. And we have, in fact, been able to find one inhibitor that does bind in the active site, right between the two aspartic acids seen here, the magnesium binding, and the glutamic acid that form the catalytic triad of this particular enzyme system. So in conclusion then, structural biology continues to play a major role in the research program of LMB and will presumably continue to do so in the future. Thank you very much.
Thank you, David. Startling, startling. I'd like to now introduce another one of NIH's premier researchers, Dr. Gary Felsenfeld, who is a world expert on chromatin structure and regulation of gene expression. And as David alluded before, a number of these gentlemen overlapped at Caltech. Gary, as you heard, received his degree at Caltech in physical chemistry and postdoc in quantum chemistry, and here he is now a world expert, as I said, in gene regulation. I think this is incredible, most amazing. Gary's, if I might share something also, Gary's interests in science, he knows what's coming, started very early. Gary was Westinghouse, high school Westinghouse awardee. Now it's the Intel Prize back in 1946 or 7 or so. And of course they come to Washington, and Gary's first introduction, remember, as a high school student, remember who he is now. He was taken to NIH and was introduced to science in the NCI, the old Building 6, where he was taken up to the attic. Remember, this is his first stimulus, where he was taken up to the attic where there were goldfish swimming about in this horrid environment of mutagens and carcinogens. But that didn't sway him at all. He continued in science, so fortunately. So Gary, please.
Thank you very much. Actually, what I said to myself then was, I'll never work here. I remember that distinctly. I said, if I have to work in a place like this, I'll never be a scientist. But when I came back some years later, it looked beautiful. It's amazing. So, yes. So my job, I think, today is to try to describe the evolution of the other half of the laboratory of molecular biology, which we don't think of as the other half, really, because they are really together. They make the laboratory. So could I have that slide, please? Thank you. This is the current makeup of the lab. These are our section chiefs. I put stars here because Wei Yang and Mike Krause recently moved from tenure track to tenured investigators, and will soon have their own sections. And the yellow boxes are the are the structural people, and the blue boxes are the people who are, quote, molecular. But the next slide is the same thing, where I've actually put a another square of counter color to indicate people who do both. So all the blue people with little yellow squares actually are tremendously interested either directly or indirectly in structure. And certainly Jim Hurley and Wei Yang have their own component of molecular biology going in their own groups. So it's really perhaps unfair to try to break it up in that way. So the next slide is to show you, I'd like to sort of give you the lineage of the people who are presently in the laboratory, and I'll start here. This is just for the people who are doing the molecular stuff. So in the early days, of course, as you heard earlier, we had Bruce Ames in the laboratory, and his interest was in regulation of gene expression in prokaryotes. In fact, he was, together with Jacob and Monod, he developed the idea of what he called coordinate repression, that is control of a series of bacterial genes by a single regulatory mechanism. And then toward the end of his stay here, he began to be interested in measurement of mutagenesis using his salmonella system, which led to the Ames test, ultimately. And he left with us Bob Martin, and then Lee Rosner, and I could also have mentioned, should have mentioned Michael Yarmolinsky, who passed through the lab, Max Gottesman, who passed through the lab, but Bob and Lee are still with us. Martin Gellert started, as you heard, with DNA gyrase, and earlier he discovered DNA ligase. But in more recent years, he's been interested in DNA recombination, antibody diversity, and things like that. And that range of interests connects with his postdoc, Kiyoshi Mizuchi, who is, of course, no longer a postdoctoral fellow, but a senior member and very distinguished member of our laboratory, and in turn, Mizuchi's postdoc, Bob Craigie, who is also a section chief in our lab. Somewhere in the middle here, we had a 14-year stay from a very distinguished Japanese molecular biologist, Junichi Tomizawa, very well known, whose interest was in DNA replication and recombination. He returned to Japan to become the director of the Institute of Genetics and has now retired. Next slide, please. Here are more of our founders and successors. You've heard, of course, about Gordon Tompkins, who really was the driving force in the creation of the laboratory and in the general good spirits, I would say, tremendous optimism that characterized us at the beginning, and I think still does. And his interest was in hormone action and, in general, regulation of gene expression in eukaryotes in the days when that was a very improbable subject in some ways, and certainly did not have the connotations it has now, which is to say, we think of this as, in some sense, a solved problem, even though we're still working at it very hard. It is in principle solved, but in those days, it was just one big black box instead of lots of little ones. And then, when I came to the lab, my interest was largely in DNA physical chemistry, and then slowly in protein-DNA interactions. And they came along about the same time, Phil Ross and Steve Zimmerman. Phil interested, and you'll hear more about their interest in a little bit. And then, I would think, to a considerable extent, it was Gordon's doing that I realized that this was really my interest in gene expression. And so I turned from studying model systems to studying chromatin structure and gene expression. And more recently, there has come to the laboratory, as our general interests have become much more biological. First, we had Alan Wolf, who came and then established his own lab at NIH, and then Mike Krause, who works in C. elegans. Todd Miles, one of our earliest people, was interested in the organic chemistry and the physical chemistry of nucleic acid structures, particularly multi-strand structures, so there was lots of interaction here. And in the middle we had a long stay from one of the world's great statistical mechanicians, Terrell Hill, whose interest was in statistical mechanics of biological systems. And he has now retired. And then you heard about Harvey Atano, one of the founders of our lab, interested in sickle cell anemia, the co-discoverer identifier of sickle cell hemoglobin, very interested in hemoglobins. And in some sense, his interests may have percolated over here to our interests because the gene expression model systems we use are globin model systems. So now I would like to start just to take you through the present status of the lab in terms of where all those interests have led us. So you heard that Martin Gellert was interested in DNA gyrase and ligase, but his present interests are in VDJ recombination, in the generation of immunoglobulin diversity and T-cell receptor gene diversity, and the development in particular of in vitro systems in which these reactions can be studied. And next slide, one of his greatest achievements in recent years has been the development of an in vitro system in which you can take a piece of DNA that's out of the immunoglobulin locus and actually go through, no, yes, no, two forward, please. Next one, please. Yes. And go through using purified enzymes, RAG1, RAG2, and a cofactor, and actually recognizing certain signal sequences in the immunoglobulin locus, you can actually then cleave the DNA specifically. These are the intermediates which he identified. And then with addition of further factors, you can create properly rejoined distant segments together with this excised region which circularizes. This is a great achievement, and he continues to study these reactions. Now, next slide, please. Kiyoshi Mizuchi, remember, started work on recombination when he was still with in Gellert's group, but his interest has been in muphage transposition as a model in general for transposition in prokaryotes and eukaryotes, and that includes retroviral integration. It turns out that all of these mechanisms are very closely related, and Kiyoshi actually developed a complete in vitro system for mu transposition, and he's worked out all the details of the reactions. So this is a piece of organic chemistry he had to do in order to work out the exact nature of the reaction by which one DNA attacks the other to insert into the host. More recently, and very characteristic of our laboratory, he's taken a completely new direction, that is to try to visualize single molecules and molecular complexes. And as is typical for our lab, I mean, this takes a while to get going. It's taken him a couple of years at least to get started, but that's fine, and I think we can be sure that interesting results will come. Next slide, please. Bob Craigie studies particularly HIV integration mechanisms. It turns out that the detailed mechanisms are very similar to the mechanisms for muphage transposition, so what we learn at one level gets applied at a higher level of complexity. This is actually something else. This is a factor identified in Craigie's laboratory. It's a cellular factor, BAF, that protects retroviral DNA from autointegration. So it keeps the retrovirus essentially from destroying itself. And this is, I've put here a structure. This happens to be an NMR structure done together with Marius Klor and Angela Gronenborn. But the same thing is being done by crystallography. And in a number of cases here, one can put a structure, and you'll see, I think, in a couple of other cases, because there are no real boundaries anymore between the molecular stuff and the structural. Next slide, please. This is Todd Miles' group, and he's been interested for years in what holds multi-strand structures together. Trying to dissect, for example, the energy contributions from base stacking and hydrogen bonding. This is a triple-stranded DNA structure solved together with David Davies' group with Sankey Rhee. And again, it's an example of how one moves. All of Todd's work has been with NMR and UV and infrared spectroscopy, but then one moves over into the structural very easily in our laboratory. And it's not rather nice to see this structure, which the first of which, as you heard earlier, goes all the way back into the 1950s, which when David Davies and I and Alex Rich showed that combining physical chemistry and structural chemistry is a very useful thing to do. Next slide. Steve Zimmerman studies intracellular crowding. So we know that, in part due to his work, that high concentrations of macromolecules, for example, in cells and in nuclei, have an enormous effect on reaction rates and equilibria. And he's interested in studying that, and also in a related subject, the structure and composition of the compacted DNA entities called nucleoids in bacteria. Next slide. Phil Ross is a thermodynamicist. He nowadays, his main interest is in microcalorimetry, and he uses that to study all kinds of trying to dissect the energies that stabilize protein structures at multi-strand nucleic acid structures. Next slide. Lee Rosner and Bob Martin are very interested in prokaryotic gene regulation. So remember, they carry on an old tradition in our laboratory. And in particular, they've been interested in the analysis of promoters that are regulated by these three proteins, Morris, Sox, and Rob. And this is, again, a structure, in this case, of MARA complex to its target site on DNA, done, again, together with the Davies group. Next slide, please. Yeah, this is our group. We keep the name Physical Chemistry Section. It's our cover. And we do some physical chemistry, actually, on protein-DNA interactions. For years, we've used the globin gene system as a model for gene expression. In chickens, we use the chicken system, and we've connected that with chromatin structure, so the interactions between regulatory elements and the higher and the packaging of the DNA, which turns out now to be a major theme in the understanding of the regulation of gene expression. For some years, we worked on higher order chromatin structure, and its physical chemistry, and on the mechanism of transcription on chromatin templates. This happens to be, again, just to keep the structural theme, is a complex of GATA1, which is an erythroid transcription factor. This is the DNA binding domain, with its target on DNA, done by NMR in collaboration, again, with Marius Klur and Angela Gronenborn, whose laboratory is the lab with which we share our building, Building 5, and we did share Building 2 as well before that. Next slide, please. And then finally, as we move into the future, we understand that we are able to deal with more complex systems. And Michael Krauss is interested in the developmental biology of the nematode C. elegans, and in particular, he's interested in the regulation of proteins involved in muscle development, MyoD family, for example, and the techniques here are quite developmental. Here we have a tagged reporter for an HLH gene, which allows visualization during development of the loci of expression of the gene. There are plenty of interactions with Michael, even though he would seem to be maybe more biological, but there's plenty of reason to think that this is the future for molecular biology. And many people in our laboratory, for example, identify genes which have equivalents in C. elegans, and the methods now exist to target inhibition or destruction of the messenger RNA produced by specific genes, and you can actually look for phenotypes in C. elegans rather quickly. So just from that practical point of view, there are plenty of people who want to talk to Michael, quite aside from the issue that these are very interesting molecular problems. So I think the next slide is the last one. It's really a recapitulation. I've put together the two slides I showed you at the beginning to show all of the molecular things that are going on here, and lots more arrows could certainly be drawn. I mean, not only the formal ones of who directly works with whom, but all of us talk to each other all the time and give each other ideas and encouragement. And off screen here are all the structural biologists whom David has talked about. And that is not a narrow passageway here. Really, it's all one big picture, and it's that synergy that makes our laboratory, made it all these years very unusual. And we think still those interactions still do make it unusual, and we hope to keep it that way. Thank you.
That's okay. I want to say first thanks to all three of our speakers for very stimulating talks. Also, I was thinking, looking at this last slide, back to something that Boom Soon said in his talk about how all these scientists working together in their laboratories are protected by 3 layers of semi-permeable membranes, the lab chief, the scientific director, and the director of the Institute from all the dreadful outside forces in politics and money and other such things. And I think the two scientific speakers have shown the kinds of intellectual pursuits that can be done given this structure. I also want to say that we're not going to take the time to make formal presentations because we've already taken the pictures, but we are presenting to each of the speakers as a thank you for coming today, a copy of the most handsome poster for this event. And this is just really a terrific poster, I think. So we are very pleased that they came and talked with us. I want to put in a plug for history before we go. If you enjoyed today's event, we would be delighted to have you join us in the History of Biomedical Research Interest Group. If you go on the NIH homepage, nih.gov/SIGS for special interest groups, and scroll down to history, it will take you to a link where you can sign up and join us and come to other presentations throughout the year. Finally, just as we get ready to adjourn, I want to again thank NIDDK for its co-sponsorship of this event and for its sponsorship of the reception you're about to enjoy. I want to thank the speakers and our 11, and thank all of you all for coming, and I hope you'll have a chance to talk with the speakers afterwards. We are adjourned.